Hereby, I open this academic ceremony in which Katrin Münzebrock will defend the academic thesis, mRNA delivery for joint and intervertebral disc regeneration. I would also like to introduce the promotion team. Welcome the promotion team here with Professor Welting, having a chair in experimental orthopedics, in particular molecular cartilage biology. Also Professor Willems, with a chair in integrated spinal care from Maastricht University. Professor Welting also from Maastricht University. And also welcome to Professor Kremers, holding a chair in experimental pain medicine at Maastricht University. Now, may I invite you to present a summary of your study and the conclusions of your thesis.
Yes, thank you. So let's start. My thesis was on osteoarthritis and chronic low back pain, which are both global issues affecting over 500 million people worldwide. And they are characterized by chronic pain and disability. And usually women are more affected or more often affected than men, which makes it also the #1 reason for reduced workforce. And both of these diseases are caused by degeneration. For the joint, it is here as an example, it's the knee joint. And here we see the changes of degeneration, mostly in the inflammation of the synovium, but also in the degraded cartilage. Because you can see, for example, we lose the smooth the smooth surface of the cartilage and also the integrity of the matrix components. And similarly, in the intervertebral disc, we lose height and also water content, which makes both cartilage and also the disc lose its function in shock absorbing and also buffering movement. Treatment nowadays is physical therapy and also pain management, and in extreme cases, surgery. However, surgery, for example, for the knee joint would mean that we remove all the damaged cartilage and replace it with an artificial knee. And in the intervertebral disc, we would try and remove the disc or immobilize it by fusing the vertebra around it. However, this means we can only treat the symptoms. And regeneration, so going back from the degraded state into the repaired and healthy state of cartilage or intervertebral disc, is not possible yet. So now comes mRNA as a new possibility. mRNA is an instruction plan for cells to build proteins. We have mRNA as the plan, and then we have the ribosomes, which are basically the workers, and the tools which then create proteins. And these proteins we can use already. So we can actually produce mRNA to use and tell our body to build proteins. A very common example of that is the corona vaccine, where we used mRNA for parts of the virus and create a very efficient vaccine. But for my purpose, so osteoarthritis and chronic low back pain, we were then wondering, can we use mRNA to instruct our cells to repair cartilage and also intervertebral disc? And here in my thesis, I have 4 research chapters. Can nanoparticles deliver mRNA to cells of cartilage and disc? Can we instruct cartilage cells to start repair in a rat model of osteoarthritis? Can ultrasound improve transport of nanoparticles into cartilage? As well as what models do we need to find new treatments for osteoarthritis? So, the first chapter. Can nanoparticles deliver mRNA to cells of cartilage and disc? It would be too easy if we could just add mRNA into the joint like that and have it work its magic. However, if we would just simply inject mRNA as it is into the joint, it would simply be degraded and it loses its function. So we need a transport system. And I looked into nanoparticles. And I was wondering if polymeric nanoparticles can be used in osteoarthritis and chronic low back pain to deliver mRNA to cells. So what we did first was to take cells out of the synovium, so the inner sleeve of the joint capsule. These were synoviocytes and also macrophages. We took cells from the cartilage, which are called chondrocytes, and we also took cells from the intervertebral disc, so annulus fibrosus cells and also nucleus pulposus cells. And what we did next was then we cultured them in 2D and we added nanoparticles, and we looked into whether they can take up the nanoparticles, so whether the nanoparticles get into the cells. And for all of these cell types, we could actually see that that's the case. But we do not only need to get them into the cells, but we also need the mRNA to be then used as a plan to make proteins. So we also checked if protein production also is occurring. And for that, we've checked with a reporter protein, which is EGFP, which if it's produced, we see that we have green cells. And this was the case for all of the cells, only not for the macrophages. And as macrophages are immune cells, we think that maybe the macrophages actually recognized the nanoparticles and the mRNA as something foreign, which they have to fight, and that's why they just degraded and broke it. So in the next chapter, because now we've seen for most of the cell types, we actually see protein production, so the mRNA works. We were wondering if we can also see this in an animal model. Can we instruct cartilage cells to start repairing a rat model of osteoarthritis? For this, we first induced osteoarthritis in a rat knee by cutting one of the ligaments and also removing part of the meniscus. And thereby we go from this healthy cartilage, which has very red matrix, so the top half, and also a very smooth surface. And over time, due to this surgery, it loses its smooth surface and also the color. And thereby we see, okay, we have an osteoarthritic model. And here we then injected therapeutic mRNA into these knees of rats. And the mRNA that we used was a therapeutic which could produce cartilage protein called lubricin. And lubricin, as the name already says, it lubricates the joint, so the cartilage surface. And then, as analysts, we then checked the knees for damage, and also we controlled for, we checked if the animals are in pain and if the treatment can improve the pain symptoms. So, going to the knee damage. Here we see the control, and if we then go and see the results, with the mRNA therapy, we still don't see the red and smooth surface, so unfortunately we didn't see any improvement here. And for the pain, what we did here was we put the rats into a cage which had like a small mat that is censoring motion and weight. And we checked for weight distribution of the paws. So we think if they are in pain, they would change the weight of the paws. And we then compared healthy to diseased, treated to untreated animals. And what we could see here is that the green line is the control, so no treatment, and the blue line is with treatment, but they are basically the same. So also here we unfortunately didn't see any improvement. It did not reduce pain. So in the next chapter, then we checked, can ultrasound improve transport of nanoparticles into cartilage? So we've seen in the previous chapter that we don't, unfortunately we don't see any improvement, so we thought, okay, the cartilage is a very dense tissue, so maybe nanoparticles, including the mRNA, get stuck in the matrix and don't reach the cells. And thereby also we don't see an effect of the mRNA. And ultrasound can actually, by acoustic streaming, push fluids into a material and also push particles into a material. So we were then wondering, can ultrasound improve transport of nanoparticles into cartilage? And for that we've tested the transport of a glowing nanoparticle into a hydrogel model of cartilage and into actual cow cartilage. And after that, we've then measured how deep these nanoparticles go into the matter and also how bright the signal is. As a sign of if it's brighter, then we also assume that more particles get inside. So for the hydrogel model, we've seen that the peaks, so the depth and also the light signal, the peaks are higher, so we see that actually there's an improved transport into the hydrogel using ultrasound compared to the control on the left. Going then also to the cow cartilage, we see that we also see similar effects, but the curves are not all showing this peak, but there's also, so there's like a tendency, but it's not as clear as in the hydrogel model. And then going forward, in the last chapter, I was investigating what models do we need to find new treatments for osteoarthritis. So you remember in the last chapter, we've mainly looked into the cartilage. However, cartilage is not an isolated tissue. We have nerves, bone, synovium, meniscus, or fat tissue, and also other ligaments, for example. But these in the joint, they actually are not isolated, but they communicate with each other. For example, via direct cell-cell contact over soluble factors or even vesicles. And this interplay of tissues should be considered if we want to study arthritis and therapies for arthritis. So I wrote a review on these co-cultures to see if there's a benefit to studying co-cultures compared to just cartilage alone. And we see that, so we studied, for example, co-culture systems of cells with other cell types, but also, for example, synovium and cartilage or other tissue components together. And in this review, we found that many studies showed an effect of co-culturing. However, we do need validation to show that this effect is also seen in patients to create valuable models. Co-culture models show potential for drug development testing, and they also might reduce animal experiments. So, in summary, nanoparticles can deliver mRNA into cells of knee joint and intervertebral disc in cell culture, and the cells can use the mRNA to produce an indicator protein, only not in macrophages. In a rat model, we could not see any knee repair after retreating with mRNA for cartilage protein, and also we did not see any improvement in pain. Ultrasound might improve transport of nanoparticles to cells in the cartilage. And co-culture models have the potential to improve studies on osteoarthritis and the development of new treatments. For the future, delivery systems need to be improved so mRNA can actually reach the intended cells. And here I think targeting is very important to reach specific tissues and cells, so not to reach all of the mentioned cells, but for example, specifically cartilage or synovium. mRNA drugs are quickly broken down, so we do need some kind of depot to continuously release mRNA drugs into the joint and have a prolonged and efficient effect. We need to study which mRNA shows most potential to produce a potent protein to stop degeneration, because now there's a lot of targets, but we kind of need to see which ones show the highest potential. And I think what's also very important is that no patient is the same. So personalization is very important, and I think with mRNA treatments, we might actually get, that is quite a good potential to use mRNA treatments for this personalization. And with this, I give the word back to the prorector.
Thank you for this presentation. We will now start the opposition, and the opposition will be opened by Professor van Griensven, Chairman of the Assessment Committee and having a chair in Regenerative Medicine at Maastricht University. The floor is yours.
Thank you very much, dear candidate. Ich habe mit großer Freude Ihre Doktorarbeit gelesen. Es war wirklich sehr schön. Und I would also like to extend, of course, my congratulations to the supervisory team. However, I do have some questions on Chapter 4. When you look at Chapter 4, you use sonication.
Yes.
And you have a model of agarose. And when you compare the agarose with the cow cartilage, the agarose was much better. Do you think that is depending on the material characteristics?
Highly esteemed professor, thank you for your question. Yes, I think it is definitely, that is one part of the why it's so much better, because we have, of course, it's much easier. It's just the hydrogel, so there's just one ingredient to it. And in cartilage, there's a lot of different factor cells, the surface. And it can also be that it might be more porous than, for example, the cartilage. So yeah, I think that might be the reason.
And did you see after the sonication, did you see then also differences when you looked at it? Did you see like pores or channels occurring in the agarose and or in the cartilage?
No, I don't think I would say there was generous, but it was mostly really at the surface, and so the highest signal was very much at the surface. So it can also be maybe that it gets like stuck there and it just doesn't get deeper, and could also be that it's that it gets stuck better in aggros, for example, and then just repelled by cartilage, and so it's
right. And when you then look at the indeed at the microscopy, you see that they are mainly in the surface.
yes.
When you use sonication, it's also important to get like your RNA in the cells.
Yes.
You use lipofectamine in this case. So why didn't you use something like nanobubbles? You also mentioned that in the introduction in chapter 4, that then like kind of explode next to your cell.
That's a good approach, but I think for us, because we've used polymeric nanoparticles before, and we thought that a similar, lipofectamine, of course, a different technique, but it might be at least similar in a way that it would be endocytosed by the cells, similar to the nanoparticles. And I think we were interested if we can improve the delivery of into cartilage for nanoparticles, and that's why we didn't choose nano.
Okay. And when you then look at that it stays at the surface, do you think that after that there is still some diffusion possible going down, so that you get like an advantage in by using that, or do you say, well, it's just at the surface, it doesn't make any difference?
You mean like using ultrasound or not?
Yes.
So I think the ultrasound itself works when it's applied. So I don't think afterwards it would get deeper. Of course, unless, for example, there's some kind of barrier on the surface of the cartilage, and with the ultrasound you could overcome that. And then it might be possible to then that it might diffuse into it later.
Because you use it on like normal cartilage. What is when you have damaged cartilage?
Yeah.
Is there then a difference? Because then the surface is already a bit more open, and perhaps, you have a different effect.
It could be possible that in damaged or osteoarthritic cartilage that you can actually see higher penetration, because we know that its porosity is higher. And of course, you also lose like this very smooth surface, and the matrix also is degenerated, so maybe the pores are bigger. So it's definitely possible, but unfortunately, we didn't have time to test it.
And when you think of using that in the clinic, so when we look at the vertebral disc,
Yes.
so I can imagine when you come from the backside. I mean, one of your promoters can say that much more better. But when you want to go to the front side, you have all the intestines in front. How do you get then your sonication there?
That's definitely very challenging. However, there is possibilities to kind of guide ultrasound in a way that it maybe even reaches only the power that it needs to, for example, move nanoparticles. I think maybe similar to like two-photon microscopy. Kind of, you need like you attack it from different sides, and that way you reach the high enough energy only in the tissue.
Okay. When I then go to chapter 3, there you use it like in the animal model, and you use also repeated dosing, but that didn't have actually an effect. So the question is that because it stays in the surface and you don't have any possibility, it's like saturated already, or what is your idea why the repeated dosing didn't work?
I think the issue could be that we also just didn't get into the cartilage at all. I mean, we didn't check where the nanoparticles end up in the end, so it could be that upon injecting they are just cleared away into the bloodstream, for example. So it's a good question, but I don't know where they end up. So
okay.
Hard to say why the replicated injection didn't have a beneficial effect.
And you also stated that the transfection per se seemed to have negative effects, right? So is it then the RNA, or are it the nanoparticles you used there?
I would hypothesize that it would probably be the nanoparticles itself, because we also seen that in the in vitro work in chapter 2 that they can have some toxic effects of higher dosing. And also it would be possible that there's some kind of immune response, because they also get taken up by macrophages. So maybe that is also why we've seen like these negative effects.
Where you now also mention chapter 2, there you actually loaded the nanoparticles not only with your mRNA, but also with an siRNA with the site 3 on it.
Yes.
So they were double loaded. That of course changes the zeta potential, it changes the size, etc. Do you think that using that is then a good method or a good model to test what is going to be used therapeutically, where you only have your mRNA actually?
So you mean combining mRNA and siRNA?
Right.
Yeah, I mean, of course, we have to consider, I think you would then have to really characterize the nanoparticles if really the size and the charge change. I think those are like the most important characteristics, especially if you want to get into cartilage. But yeah.
Do you even consider an siRNA that is then effective? Because here you just used it to show where your transfection was, but do you even consider using a functional siRNA?
I mean, we didn't test that, but I think in the future it might also be a good idea to use siRNA or mRNA or any kind of nucleic acid, because I think at least for the polymeric nanoparticles that we've used, they should be suitable for any of those nucleic acids.
Okay, I'm happy with the questions and I give back to the pro-rector.
The opposition will now be continued by Professor Van Os. She was a member of the assessment committee, having a chair in connective tissue regeneration at Erasmus MC in Rotterdam. She's present online. The University of Maastricht appreciates very much the presence of external experts, and we would like you to start the opposition, or to continue the opposition.
Thank you very much, Mr. Rector. Dear candidate, sorry I cannot be there today, but I have read your work with very great interest. I've been following this research line that now moved from Utrecht to Maastricht for quite a while, and I'd like to congratulate you as well as the team that supervised you with these next steps that you made to get it closer to application. So my question is, or my questions, I have to say, are about targeting the macrophages. And I would like to start with chapter 2, where you show that the macrophages take up the nanoparticles, but they do not express the mRNA. And but there seems to be an effect on proliferation, which suggests a pro-inflammatory effect, maybe. So you did these experiments on M1 macrophages, I believe. Did you ever try to do it on other type of macrophages? And would you expect potentially a different response?
Highly esteemed opponent, thank you very much for your compliments and also the question. I think so. We did not test it on the other macrophages, so we only tested it on M1-like macrophages. Yeah.
And if you would hypothesize, do you think
okay,
the other macrophages might respond differently? Would also take it up?
Yeah, I would assume they also would take it up. Yeah, because the uptake mechanisms I think are very similar. But I'm not sure how they would react to it. It can also be that because M2 and M1 are of course not like very like separate, but they also can, you can go from N1 to M2 and the other way around. It's like a spectrum, right? So it can also be that upon taking up or having contact with the nanoparticles, the polarization might also change. So it can also be that even if we try to transfect M2 macrophages, we end up having M1-like macrophages, or the other way around. But I didn't test.
You didn't test it?
Yes, I didn't test it.
Yeah. So this potential pro-inflammatory effect that you saw in chapter 2, I think you also see it in chapter 3 when you used when you injected them in an inflamed joint. So when I look at figure 5b of chapter 3, so that's on page 58, you can actually clearly see that it has an effect on inflammation in the group that got sham operation and injected, and where you injected the scramble.
Yes.
Yeah. So do you think this is due to the fact that there was already some inflammation going on? So have you ever injected them in a healthy joint? Or, I don't know, one of the people that worked on the group, in the group before you, maybe?
So unfortunately, we didn't we didn't inject into healthy. So we missed this control here, that we can actually compare whether the nanoparticles or the sham surgery induced these changes.
Yeah.
Yeah.
Yeah, it makes sense, right? That you didn't do that because you would not treat a healthy joint.
Yes.
And you always want to have as little animals as possible.
Yes.
But assume you would have done it. What would you have expected? Would you expect the same result?
So the thing, I mean, we've seen like some reaction to already we've seen that they can be taken up by macrophages, right? So I would also assume that even in the healthy joint, you would also see some uptake. However, whether we would see like a higher Kren score and higher inflammation, I'm not sure, to be honest.
Yeah, yeah. So yeah, because I was thinking maybe you should exclude very inflamed joints for this therapy. So what do you think about that statement?
To exclude what again?
So now you injected it in a very inflamed joint, even at day 7 when the inflammation is at the highest level. But maybe because it seems you get a little bit more inflammation even when you do that, maybe you should not inject it in a very inflamed joint.
That's a very good suggestion. I think that might be possible. I mean, there's of course very different colors, let's say, of osteoarthritis, how it looks like. There's more inflamed phenotypes or more cartilage-based phenotypes, and it could be that if we already inject in inflamed joints, that there's already so many immune cells that we just trigger even more an immune response if the nanoparticles actually did that, but we can unfortunately not really prove that.
So of course you didn't look at that, but assume you would like to look at that, because otherwise now it doesn't look so promising, but it might be. So then which model would you use? You have a whole bunch of models in chapter 5. Would you use one of these, or would you actually go to a different in vivo model or a different time point in the same model?
I think it would be, I think if you really want to test the reaction to synovium, I would assume it would be best to either have maybe like an explant co-culture, maybe even with synovium and cartilage, or have a similar model that is less inflammatory, or maybe an earlier time point in animals, even rats. Because if you would do it in rats and have like a less severe immune response, you could also nicely compare it to the results that we already have. Or if you would use it in the co-culture system, there you could also vary, like if you use, for example, healthy-looking synovium or degraded synovium or inflammatory synovium, and also you could vary if you have more healthy or more degraded cartilage. So you could even like mix and match basically, and try and at least model different, even different types of osteoarthritis, more inflammatory or more cartilage-based osteoarthritis models.
Yeah. Very interesting. I hope your supervisors actually took notes, so that this can be done afterwards. I'm very happy with the answers. I'll give the word back to the director. Thank you very much.
Thank you. And the opposition will now be continued by Professor van Zundert, also member of the assessment committee, holding a chair in pain medicine at Maastricht University.
Thank you. Gratulieren. It's my only German word that I know, I think. But it was very interesting to read. It was outside of my comfort zone, so I learned a lot. Thank you for that. So I'm a clinician, so I will a little bit discuss with you from a little helicopter view. But can I ask first one of your paranyms to read proposition 8? At least that I counted 8. It's about the vaccine process.
mRNA therapies pose a great opportunity for personalized healthcare and future, not just the vaccination purpose.
Thank you. Personalized healthcare is will be very important in the future. We all hope on that. But how do you see it in the future? When we can apply that? When will it be safe? Are we yet there?
Unfortunately, I think, oh, wait, highly esteemed opponent, thank you very much for the compliment and the question. I think we're not there yet. There's like different hurdles that we still need to take. I think one big issue is like the legislation issue, because we didn't have mRNA or gene therapy-based therapies before. And because they get like the thing is that we have, for example, we can legalize, for example, like a transport to like the LMPs that were also used for the vaccine. But I think for now it's a bit difficult to then, whether you have to legalize like every single payload or if it's like one fits all. And I think that's like one barrier that we still need to, but that's like bureaucracy. And then on the other hand, I mean, we still cannot really reach the specific tissues. I mean, targeting liver, for example, is very easy. But if you talk about osteoarthritis and chronic low back pain, it's still quite challenging to actually get into specific tissues. So I think I will not put like a date to it yet, but I hope it will be in the future, but it's not going to be in the next 10 years.
But it's the work has to continue. So you did a lot. You did now first part of basic work. You mentioned a lot of the hurdles that you observed and that you challenged, which is always the case with research. But now we have to find a bridge to bridge the gap to the clinic and to the patients. You describe it quite well in your impact paragraph, page 120 and 21. I enjoyed reading it. But we have to go to the next stage. So you have done now your PhD. Probably you will apply for a postdoc position. There will be some investors with research grants. What's your best strategy? What is your best way to go now?
I think what really is important is to get these transplants to really reach the intended tissue or cell. So, for example, in osteoarthritis, if we want to treat like a cartilage-based osteoarthritis, where we see mostly the degradation, I think it's important to have a transporter that we...
I'm interested more in low back pain at this moment. It's a little bit more complex. It's a little bit more challenging. So what will be your best approach for the next step in the research?
I think similar here, you would like to actually reach the disc, of course, which is maybe even more challenging because the knee joint, it's more or less easier to inject there, but the spine is, of course, a bit more, not fragile, but, well, risky, let's say. So I think that's going to be also challenging. I mean, the thing is also, I mean, ideally, there would be like some kind of solution where you would maybe inject into the bloodstream and then it finds its way somehow into the disc, but because there's no blood vessels there, it's very challenging to...
The investors are interested in results. When will you deliver? What will you deliver in the next years?
Okay. I haven't really dealt with investors yet.
Yeah. Low back pain is multifactorial. It's complex. So many people are suffering from that.
I mean, what we can try is not just tackle the disc itself, but also the nerves and the nervous tissue, because there's not only it's not always the case that there's actually the degradation or that is the cause of the pain, but it can also be neuropathic pain, for example. And we can try and tackle that. And it might be that it's a bit easier to reach those, but it's still very challenging.
Okay, now it's becoming interesting. You mentioned already in the discussion already that you want to stratify. It is also one of your other propositions. I will not ask again one of your but you mentioned also endotyping as one of the future approaches to improve the research. Can you explain it a little bit? Can you elaborate on it?
Yeah, so endotyping is to basically look at molecular markers in patients and see if you can group them based on the molecular markers. Because for now, it's mainly we look at the symptoms that the patient shows. For example, if they are in pain, how immobile they are, or we take like a check maybe how much inflammation is there in the joint. But if you take molecular markers, you can maybe more specifically get to the reason to why we see the degradation or the inflammation. And I think grouping then means that we can actually look at the specific patient groups more closely and even find solutions that might fit better to this specific group. And especially if you, for example, want to use, if you do like a clinical trial, I think if you have, for example, a lot of like osteoarthritis patients which have maybe the same symptoms, but molecularly they are very diverse. And if you would simply test one drug on all of those, I think it's very difficult to then get like a common result. But if you stratify and group them based on these molecular markers, I think that then you might, for one specific group, also get better or more promising or clearer results, let's say, with less variability.
Now we're coming back to the personalized medicine, where we started this little chat. I'm happy. Thank you very much.
That's always good to be happy. The opposition will now be continued by Professor Trifonidou, member of the assessment committee, holding a chair in regenerative orthopedics at Utrecht UMC. I thank you on behalf of the University of Maastricht to take all the effort to make the trip from Utrecht to Maastricht and to have the opposition.
Thank you, Mr. Prorector. The trip was safe, and I'm here to discuss with you a lot of questions which we have not discussed before. I always want to surprise you in that. So I would like, dear candidate, to discuss with you first on chapter 3, and where you studied PRG4. And I was wondering, who produces PRG4 in the joint?
Highly esteemed opponent, thank you for the question. There's not only one cell type that produces PRG4. I mean, we focused on the cartilage, but there's also, for example, synoviocytes that can produce it. Yeah. So we might also tackle those or target those.
I mean, you're trying so hard to get cartilage cells to produce PRG4 with mRNA delivery, but what if the synovial cells would produce it? Would that be sufficient?
It might be. I mean, it can. The thing is that if it's then secreted and can also get into the synovial fluid and maybe then be integrated into the cartilage, it might be enough. Yeah. But I honestly don't know whether that actually happens. So
in essence, synovial fluid is a hyperfiltrate of all the proteins that pass by. So theoretically, that could be possible. Yeah. And I was reading your results, and I got intrigued by the bad results. I mean, it's poor results always put you more into thinking. Yes. And I wanted to explore with you a different theory of why you see bad results. And so there is no good or bad answer in that. So because you say, oh, okay, it's probably the nanoparticles that did something bad. What if I would put a statement in your list, like you always also need to know how good the product is that you got, because there might be endotoxins in the product causing actually inflammation. What do you know of the particles that you used of?
I mean, they were produced by a company, so I didn't produce them myself. So I don't have like a direct quality control whether we have any endotoxins or any other.
Was that was that checked, or do you know?
I'm not sure, to be honest.
Okay. Because it can be, if it's not GMP produced, that they have endotoxins in there. And there is a beautiful model where you inject very little bit of endotoxins in a joint and you get massive inflammation. Okay. So when I was looking at your histology, I was thinking, could that be the reason for the poor results? If that would be a question I had posed 2 years ago when you were doing those studies, what would you do?
I think we would have to take a closer look at the nanoparticle product that we got and either check ourselves for endotoxins or or the company would do that. And then we could, of course, also, if we know that that can happen and if it's not GMP produced, for example, I think it would also be good to then include maybe like some kind of endotoxin control where you have not the nanoparticles, but just like the endotoxins that we found and test them, for example, inject them in the knee joint and see if that shows the same results or if, in fact, they might not matter that much and it is actually the nanoparticle itself.
And if we look at figure 5a, where you have the Menking and a Crane score, is there any argument you could pose that it's, Professor Trifonidou, I don't think it's only endotoxin? Comparing scrambled, OA, scrambled, sham, BRG4, OA.
I think it's not that easy to say it from these results.
Why do you think so?
Because, I mean, we have the scrambled control where or the sham and scrambled, so it's like two controls at once and not separate. So we also don't know whether it's like just the sham surgery that did this. So I think in that case we would need to have one control. We have only the sham surgery and no injection of nanoparticles, and then even maybe a healthy joint and then inject in the nanoparticles.
Yeah. Because if the endotoxins were somewhere in the production, they would end up with all the particle combinations that you take.
Yes.
Yeah. So taking this a step further, I was looking at your cartilage experiment results, where you used human donors to see whether there was a beneficial effect. And you're talking about GAG in the media. The glycosaminoglycans you find in the media, what do you think are they? Are they released, produced, degraded from the matrix being present? How do you interpret them if they're present in the media?
Let me just see.
You talk about this in page 53, and the results are in figure 355.
Yes. I think the thing is that we don't exactly know, because we just measure the total amount of GAG released, so we don't know if they are produced upon the PRG4 treatment or if it's triggering like a breakdown, and that's why it's released.
And because you use now one specific time point to look at, and if I counted well, I don't remember exactly the details, but I think you looked at day 6, and your pellets were for 7 days treated with TGF-beta, and then you added IL-1-beta. Can you help me with the design details?
Yeah, so first, we've pelleted, then we had TGF-beta for 1 week, and then upon treatment with the nanoparticles, we also added the IL-1-beta.
Yeah.
Only then.
So, theoretically, if we would have then collected a sample at the starting moment before you give IL-1-beta, you would have an idea of what the GAG content is.
True. Yeah.
And then monitoring over time, we could perhaps decipher whether it's release, degradation, production?
Yeah, I think so. At least we would, so if you don't trigger with like this inflammation or with the IL-1-beta, I can imagine that we might at least have a comparison of what the IL-1-beta does compared to not. I think then we also would need some kind of control where we don't have the inflammatory environment and also add the nanoparticles there and see if we see an increased GAG, and that might hint at that we actually see production. But if that leaves it unchanged, then it can still be that it's because of degradation of the pellets matrix.
Yeah. So I see, obviously, if I would give you a waiver of €100,000, you would have a lot of questions.
Yes.
Yeah. And you plot also GAG per DNA. And I was wondering, why do you do that? What do you want to convey?
So GAG per DNA, here we wanted to check, because I mean, the pipetting is by hand, so then it might be that there's like a bit less or more cells in the well, and that therefore the pellet just starts with more or less cells. So that's like a way to control or to normalize that we know, okay, because the DNA also, of course, gives an indication of how many cells there are, and that way we can kind of control that it's...
The DNA is on day 6, right? 7 plus 6.
Yes.
Yes. And that may change also over time.
Yes, also true.
So what does it say, a gag per DNA at day 13 of an
I
experiment?
think it still would
I
say
see
like
the
how
rector
much-
is going to save you from the answer, but give you a short answer and then I'll give it back.
I mean, it still can give an indication of how many cells there are. It can also, if we see that the DNA increases upon like PRG4 transfection, it can also indicate that maybe we've even induced cell proliferation.
Thank you very much.
You're welcome.
Thank you. Now the opposition will be continued by additional opponent, Professor Van Rijt, also secretary of this committee, holding a chair in inorganic nanomaterials for regenerative medicine at Maastricht University.
Thank you, Prorector. Dear candidate, first let me congratulate you on finishing your thesis. As a nanoparticle scientist, it was very interesting to read. And I also want to extend my congratulations to the supervisory team. But we're also here for some questions, so I have some additional questions. And I want to actually first start with a more overall general question. So in the last slide, or one of the last slides of your presentation, you say some things about future outlook of what should be optimized. And you mentioned actually two things, a delivery platform, but also selecting the therapeutic target. So my question is actually for the RNA-based therapies that you are exploring for this specific application, what do you think is ultimately more critical, optimizing the delivery platform or selecting the therapeutic target?
Highly esteemed opponent, thank you for the compliment and also for the question. I think for starting, I think it would be most important to have something that really very well delivered, or where we can have good targeting and also like a continuous effect in the joint, for example. Because I think unless we have the ways to get there, it doesn't matter which target we have.
Yeah, I completely agree with you. Sets me up for the next question, actually, which is also kind of related to previous opponent. I want to actually explore with you another alternative for why your in vivo experiments did not work. And so this is mainly related to chapter 3, but I think in general. So bear with me. My first question is actually about your in vitro results. So in this chapter 3, you treat a pellet with nanoparticles, all right? And you show that increased GAG production compared to the buffer control. However, the PRG4 mRNA was often not superior to the EGFP mRNA control. Yes. How do you interpret that finding?
So I think it might be due to the fact that maybe the nanoparticle itself can actually trigger this maybe GAG production or release, or it could also be that if there's an effect of the nanoparticles and it just is so big that we don't see the effect of the PRG4 anymore. We cannot exclude that there's no effect of the PRG4, but it could be that the nanoparticle itself somehow triggers this change even more compared to the mRNA that we are using.
And what additional controls, so I agree with you, but what additional controls could you have included in your experiments to actually disentangle these nanoparticle effects?
I mean, we could have used, for example, a different delivery method and checked if we thereby can improve, or thereby check if it is the nanoparticle itself that does this. Alternatively, maybe we can even try and just treat directly with the proteins. However, I'm not sure how well that works, but that would also be an option.
And what about just including empty nanoparticles?
Yeah, that also would be a good. Yeah, that also would be an idea. Yes.
So my next question is actually related to also chapter 2 in vitro results. So here. Actually, I wanted to ask, in the 3D pellet model, did you also verify EGFP expression or otherwise confirm that the mRNA cargo was successfully delivered and translated within the pellets? I couldn't see that in
Yes, I believe we did that, and we could see some EGFP translation, at least on the surface of the pellets. So we do see that there's at least some of the surface, but it could be, of course, that we don't reach the core of the pellet. So that can also be that because we only have the treatment at the surface, that thereby we also don't see the full potential of the nanoparticles.
Right. So then, do you think it's possible that the PRG4 mRNA is actually not working within the pellet system that you use, and how could you check that?
I mean, we can of course also check simply first in a 2D culture model, like in chondrocytes itself.
Yeah.
Or what we could also do is the first transfect and only afterwards make the pellets from the cells.
Yeah.
So then you would definitely know they had the opportunity to get to the cells, and if we then see an effect, then we know whether or not the PRG4 mRNA works or not.
Yeah. Yeah, so in my view, I think there's some critical steps into really evaluating whether your mRNA PRG4 actually works before going into in vivo. And that actually, I don't know if I have more time. It leads me to a more general question about the nanoparticles, but I also understand this was not something that you developed. But in your chapter 2, I found that you had quite a high polydispersity index of around 0.34. We consider that quite high. And my question was actually, do you think that this heterogeneity might influence your results? And if yes, how?
I think it could definitely, polydispersity also means that you cannot get like the same batch, right? So again, also imagine that by chance you just get like a batch where there's higher like bigger nanoparticles or smaller nanoparticles, and if you don't control for that, it can also be that of course also influences the results that you're getting.
Yeah, for sure.
Yeah.
For sure. Okay, I'm happy with the answers. I'll give the floor back to the prorector. Thank you.
Okay, now we will continue the opposition with Dr. Emans, member of the assessment committee and orthopedic surgeon at Maastricht University Medical Center.
Thank you very much, Prorector. First, my compliments to you and the team. I think it's quite challenging to take both osteoarthritis of the knee and back pain to really change the life of patients, or at least to start with that. So as you can expect, my questions are, of course, about the clinical implication and introduction of your thesis, together with my colleague. He, of course, about the spine, and I will do it for the knee. So I would like to use this last part of your defense to see how we can get it into the clinic. So my question is, first of all, about the chapter with the ultrasounds. You had several questions already, taking the agarose, doing other things, but I saw you took animal cartilage, and was it healthy or was it diseased cartilage?
Highly esteemed opponent, thank you for the compliment and also for the question. So in our experiments, we've used healthy bovine cartilage, yes.
And what would you expect? Because we see that the matrix is quite dense of healthy cartilage, but what is the case in degenerated cartilage? So and do we need to ultrasound in degenerated cartilage?
So the matrix changes, of course, with the degradation. So there's a bigger pore size, and also we lose like this surface. So it can definitely be that it actually penetration improves if you have degeneration. So I think also if we actually want to have the ultrasound, if we want to use the ultrasound in osteoarthritis, I think it also is definitely important to also test not only healthy but also degraded cartilage. And then we could use, for example, like a model of bovine, but because also at least at the UMC we also have access to patient material, I think it would even be better to look at the patient material itself.
Thank you very much for your answer. So when I compare to my colleague, with these answers, I can help you maybe finding the investors. What we also need is that we need to see what happens if the particles are in the cartilage, let's say degenerated cartilage. So if it's degenerated cartilage, then macrophages can follow them as well, probably.
That depends on the pore size, but it can be, yeah.
And what does a macrophage do with a foreign body? And what's it being secreted? And how will then the cartilage respond, tissue respond on that? Can you elaborate a little bit on that one? Is it negative that they, or will they remain in the cartilage and nothing happens? or will they give a reaction when the cartilage starts degenerating because macrophages attack the release system? So I'm searching what cells will be there and what the body will do.
So if it's really the case that macrophages can also enter the degraded cartilage, and we might even improve this upon ultrasound stimulation, then I can also imagine that that can have bad effects, because we've seen already that nanoparticles can be taken up by macrophages, so it might be, and macrophages generally are like cells that like to endocytose. So it can be that we simply get the macrophages end up taking everything up, and we don't have, at least we don't have on one point the beneficial effects of the nanoparticles in the chondrocytes. And if it's actually the case that the nanoparticles also trigger an immune response upon uptake into the macrophages, it can be that also we then see worsening, that we have actually secretion, for example, of inflammatory markers that then continue to break down the cartilage. So it can be definitely detrimental, yeah.
Thank you for your answer again. So I totally agree. That's an open thing. I would imagine having foreign bodies in a body can be a danger of these processes, right? So you solve maybe one of those things to look at it, but how can we really find it? If I look at your model in chapter 5, how would you then do this experiment? Because you have this different systems, you might have macrophages, and you might get degenerated cartilage where with the ultrasound you can put in, or even without the ultrasound. So can you elaborate how you would make such a, if you have macrophages coming maybe from Hoffa's fat pad, you have cartilage, you have your particles, how can you visualize this process, or can you look at it, and can you elaborate a little bit how you would like to do that experiment?
So I think for that an explant co-culture would make sense, to have either like healthy or degenerated cartilage, and then add, for example, macrophages to it and the ultrasound, and see, for example, with if you have like a cell tag on the macrophages, you could then treat with the ultrasound and see if the macrophages get inside, and also see if the ultrasound improves or increases the uptake of a macrophage, or moving into the macrophages into the cartilage. And then you, for example, could, if it's like bovine cartilage or human cartilage, it actually doesn't matter. You could take slices and image and see if we see macrophages inside the cartilage, and if upon nanoparticle, or if upon ultrasound treatment we even see more, or maybe even less, or just a change generally of the uptake of macrophage into the cartilage.
Thank you very much for your answer. I see one of your mentors taking notes, so I think it's very good. Same as with my colleague. Then finally, if we want to translate that to the clinic, let's say we take the steps what you did. You start in vitro with cells, then you try to elaborate that on pieces of cartilage with the ultrasound, and finally you go to an ex vivo model, which you described in chapter 5. And then I still have a little bit of a problem, because your model of the mice, they didn't support any pain. It was rather difficult to do the histology. My question is, you took the example of using this technology for liver diseases, which they use mRNA, or vaccines. Can you tell me what the big difference is between those kind of applications and the one we have? So as a readout, if it works, I can help a little bit.
So comparing like liver or...
Liver, so you use technology and it's successful for vaccines, right? Yes. But we always struggle to find a treatment for osteoarthritis. And I think the struggle is in the readout. So the readout is pain, or the readout is histology. Can you elaborate a little bit what the readout is when you use it for a vaccination, and is it a clinical reported outcome by patients that they feel better after getting the vaccine, or what happens? What's the readout in vaccines and liver? Why successful, and how does it is it different from what our readout is?
Yeah, so I think for vaccines, the huge benefit is that you basically just need a small amount and you just trigger protein production of the virus.
You may finish your answer or not.
I'm fine finishing it. And there then later on, so we just need like a small amount of the protein, and you easily reach the cells at the in the muscle, like the immune cells. And for our readout, we would need also continuous production, and the effect is not by the immune system that basically creates the immune response, but in osteoarthritis or chronic low back pain, you would need like a prolonged effect, because I can imagine that it takes much longer to repair cartilage or intervertebral disc compared to simply producing a virus protein.
Thank you for your question. I hand back the word to the...
Katrin Münzebrock, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and of your defense. I request that you and your company await the results of our deliberations and our return in this room.
Maybe they shut it off, like the...
This is your one. Yes. Super nice. Please be seated.
Katrin Münzebrock. The degree committee here present has assessed the quality of your thesis and of your defense. In view of its positive verdict and taking into account the previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Welting is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Katrin, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
That is very good. Then, by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Katrin Agnes Münzebrock, the degree of doctor and grant you all rights attached by custom and law. And as evidence of this, I now present to you the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of our university.
So now the honor is all mine. Zeer geëerde vrouw, very learned lady, zeer geleerde vrouw Münzebrock, lieve Katrin. I will continue in English, as my German is not my forte, and I guess Dutch, although we discussed maybe Dutch would have been better for you, but anyway, it will be in English. So Katrin, you did it. And with the 200% so common to you. Every PhD journey is a special one for the candidate and the daily supervisor, and yours has been far from an exception of that. Your trajectory started with a Teams meeting in COVID time, where you presented your master thesis project. That presentation and the energy that you used to present it and was radiating from the screen were convincing reasons for getting you on board of the Carthago project that was recently granted. I was soon surprised how swiftly you arranged for housing by yourself well before the start of your contract, and you turned out to have been kid-sitting there even before holding a pipette. As the pandemic was still limiting our lab time, in particular mine, so indeed you didn't see me for a long while, at least not in person, you started with a review on co-cultures for joint tissues, an important subject in these times calling for a reduction in animal use. And then together with Adriano, you started embarking on the characterization of the polymeric nanoparticles from 20-med for application of messenger RNA delivery in osteoarthritis and disc degeneration. You very systematically carried out this for all cells in the joint and the intervertebral disc, concluding that in 2D cell culture, as you already explained, these particles were very efficient tools for transfections, but not for macrophages. Pretty stubborn. Unfortunately, things didn't work out in vivo that much. That was a bit more of a challenge, as you also explained, but that's just the way that things work in science. And I'm happy we're still going to try to publish this because it's also important to publish negative data. In the course of your visit to the group of Heikki Nieminen in Finland, you embarked as one of the first scientists ever on a project to use ultrasound to enhance penetration of nanoparticles in cartilage. I think you did not really, emphasize that, but it was a very challenging and very new project, and I think you did really well in trying to set up things also at the UMCU back with all these agarose gels. And all of these things you undertook with the energy and positivity that is so characteristic of you. From your famous Friday afternoon experiments on clearing of tissue, I don't think I saw many data on that project, including G's apples, to making snowmen, that first picture I have, cutting your hair for wig making and letting it grow again, cycling all the way from Leidse Rijn to the Uithof and back on pure muscle power, no e-bikes here, and learning to speak Dutch and being an enthusiastic part of a soccer team. All was done at 200% energy and conviction. And when of that 200% suddenly nothing was left, you picked up the pieces step by step, at a rate you entirely controlled yourself, which I think has not decreased but increased your strength, and hopefully also the confidence in yourself. It should. At the basis of your powers, I think, is your capacity to be a friend, not in the least starting with your family in Germany, of which I heard so many stories over these years, you wouldn't believe it. Finally, happy to meet some of you at least. But you also got along really well with the members of the consortium. And I think your parents instilling the importance of a good heart was clearly successful during your scientific bicycle tour in the Netherlands. So you're still in touch with Paola, understood on a regular basis, and with Carla from the consortium, and even with the people you stayed with in Nieuwegein at the onset of your trip here. And in the lab, you were always someone who would be willing to help in experiments, aliquoting, whatever. You took the lead in organizing the group's labs duty and the materials we had. You asked a lot of questions. I realized you were always the one who had questions at whatever meeting we had, also internally. So I think that also shows your energy and your interest in what other people are doing. The only time you were really overdoing your kindness was when you know the answer, when you were labeling the Eppendorfs for your own students. I hope you can promise me never to do that again.
Not gonna do that, yeah.
Good. Katrin, I really enjoyed working with you, both from a scientific perspective, but also for your gentle team spirit, with every now and then this bubbly love that we saw. Be as proud of yourself as I and undoubtedly everyone around you is, and go and enjoy this day. I'm sure you will have a fruitful career in life ahead of you, wherever you land. And for now, congratulations.
Thank you.
And I give the word back.
Dear Dr. Münzebrock, also on behalf of the Maastricht University, I congratulate you und congratuliere deine Familie with the degree you have acquired. With that, I close this ceremony. Eventually your family can join or whatever. We will see how it goes.